Now, I'm delighted to welcome our next speaker, Zane Libke. Zane is a researcher and educator with a passion for making science accessible to everyone. After receiving his bachelor's degree from Davidson College in 2021, Zane moved to Ecuador to start a field genetics lab at Sumac Cauce In-situ. His research focuses on better understanding cryptic diversity in the world's largest vertebrate genus, Pristomantis frogs, as well as simplifying low-cost genetic sequencing labs. Zane also collaborates with the Mauricio Ortega Lab at the University of Iquiam to conduct field genetic courses and develop interactive teaching methods and materials. His talk title, Fungal Sequencing Reveals Cryptic Amphibian Diversity and Builds Local Research Capacity in the Ecuadorian Amazon.
I was 8 years old when I caught my first snake. I still remember this moment like it was yesterday. I was on a canoe trip with my family, and I saw this little creature dash off into the water, and I jumped into the water after it, grabbed it by the tail and pulled it out, and held it up happily for this photo. And I remember looking at this creature in the eyes and just being filled with this fascination. What kind of secrets does this creature hold? And it was that same fascination that led me to the jungles of Ecuador. My first time, I was a college student, and I did a month-long project on what first started out as snake diversity, but then I couldn't find any snakes, but I found a lot of frogs. So I spent a lot of nights going out into the jungle with a headlamp and looking for frogs, and I was amazed by what I found. An incredible diversity in frogs of all shapes and sizes. And this area that I'm working in, the Yanganates-Sangay ecological corridor in Ecuador, has an incredible biodiversity of species. Current counts stand around 140 species of mammals, 105 species of reptiles, over 700 species of birds, 149 and counting species of amphibians, and many more that are likely waiting to be discovered. And so this area is not what you typically think of when you think of the Amazon. It's actually an incredibly mountainous area that experiences some of the heaviest rainfall in all of Ecuador and all of the world. And so we're located right in between two massive national parks, the Parque Nacional Sangay and Yanganates, and there's an amazing network of researchers and conservation activists working in this area to try and better understand what's going on. And so after I returned from my first trip to Ecuador, I read a paper that really completely revolutionized the way that I thought about how we could study biodiversity. And it was then that I took the leap and moved to Ecuador and started the Sumac Cauce In-situ Genomics Lab. And so this right here, more or less, is all of the equipment that we use to do our research. You can see the mini PCR and of course, the MinION, and the backdrop of the beautiful Yanganates National Park. And so this is the lab. It's actually the product of the pandemic. The founder of the lab, Henry Sanchez, along with some other people, constructed it during the pandemic while they were stuck out in the middle of the rainforest with nothing to do. They thought, what better than construct a lab? And so you can see right here this little solar panel. That little solar panel powers all of our research. So this is the inside. We have a lot of didactic material, and it's a great space for teaching and also research. And so this protocol that we use is adapted from the protocol here on the top, and basically we really try and simplify everything as much as possible because sadly in conservation, funding is really hard to come by, so we have to squeeze every penny we possibly can and make the best out of it. So our extraction, for example, costs 7 cents. And then what we're doing right now is generating barcode sequences of as many species as possible and then creating consensuses using NG species ID. And so here's an example of one of our projects. So we did an expedition to 2 different mountains on either side of the corridor. You can see right on this map, there's a river that goes in between these two areas, and So we conducted two expeditions around 7 days each, and we found two different but similar frogs. And we went back to the lab, and we sequenced 16S and compared them. And turns out both of these species didn't have a hit. One, they didn't have a hit to the BLAST database, around 92% similarity, suggesting that likely they might represent some sort of undescribed species. And then we compared them to each other, and they don't compare to each other either. So likely these represent two undescribed species living on opposite sides of the Pastaza River that are in need of conservation. And so this information is what we pass along to our collaborators at the Ecominga Foundation, who work actively in buying land and protecting land in the area. And now with this kind of resolution of species, they're able to inform conservation decisions. So this is something we've done to date with around 45 different species, and the results are remarkable. The diversity is unprecedented. We found that out of 44 different morphospecies, we've identified 18 as potential candidate species. And these are species, keep in mind, that we know next to nothing about. If we don't even know what species they are, how can we know what kind of compounds exist in their skin that could be of use to medicine? Or how could we know how to best conserve them? And so this is really important work that we want to continue doing. And so, as you know, the Nanopore is a really diverse device, and it allows us to do a lot of different things, including endangered species ID, eDNA, fungal long-read barcoding, and soil bacteria metagenomics. And so, like I said, so far we've really only looked at the species that we know, the communities that we know. This doesn't even take into account communities that could be so hyper-diverse. And so there's a need to really create more labs like what we're doing and capacitate more research to do the same thing. So that's exactly what we did. We started conducting field genetics courses, around 3 to 7 days of intensive field, lab, and bioinformatics, and it's been a real success. We've so far capacitated over 70 Ecuadorian students, and we really need more because there's a huge demand and there's a need for this kind of research. So here's some photos from our courses. You can see loading a flow cell on the top. Really important practice. I'm sure many of us here have busted a flow cell before, so it's nice to get some practice before you try it on the real thing. And of course, these courses are really fun because a lot of ideas and collaborations come out of it. So we've had a lot of students who want to come back and want to do more in-depth projects. And so we're working to try and establish those opportunities so that students can do that same thing. And so these are just some things that we've learned over our time establishing this project, things that are needed, and we'll touch on this more on the plenary talk. Lastly, this is a team effort. This is something that could not be done without the collaboration of many, many different actors, both internationally and especially on field in Ecuador. And especially want to thank Henry Sanchez, the founder of Sumac Causa In-situ, who had a vision early on, and Walter Killenbacking, Grace Reyes, and Mauricio Ortega for their continued collaboration and expertise in organizing and instructing these courses. And there's one last thing that I want to leave you with. That conservation is not a work that ends in one generation. Conservation is a work that impacts the next 300,000 years. And so the most fulfilling thing for me from this whole work, all of the work that you see here, is when I'm able to pass on that same fascination that I had when I saw that snake to the next generation. Thank you.
Wow, thank you, Zane.